Welcome to Biomanufacturing and Fermentation Technology. This is your monthly bioprocess and biomanufacturing intelligence, the data, the decisions, and the scale-up realities shaping what comes next.
Welcome back to the Deep Dive. It is Friday, January 30th, 2026.
Happy Friday.
And we are digging into the monthly Bioprocess and Biomanufacturing Intelligence Bulletin for this past month. And I had to say, as I was going through the table of contents, there's this real tension in the air. On one hand, you've got these headlines that, they feel like they were just ripped from a hard sci-fi novel. We're talking synthetic genomes, literal space factories, AI driving evolution.
Stuff we dreamed about a decade ago, yeah.
Exactly. And then on the other hand, you have the very grounded, very gritty reality of industrial capacity, of regulatory compliance, of patent cards. It's this wild mix.
That tension is exactly where the industry lives right now. You've perfectly described it. It's the friction between what is biologically possible and, what is actually economically viable.
Right. Can you make money on it?
Can you make money and can you get it approved? And January 2026, it really feels like a tipping point. It feels like those two worlds are finally starting to speak the same language.
But the translation is still a bit clunky.
The translation process is expensive. Very expensive.
It is. And that's really the mission for this deep dive. We want to unpack that friction. We're not just going to list the news headlines. We need to understand the structural shifts happening underneath all this.
The why behind the what.
Precisely. We're talking about the completion of the SE 2.0 project, the synthetic yeast genome. This has been a decade in the making.
A monumental effort.
And then we're looking at something called T7 Oracle, which claims, and this number is just staggering, it claims to accelerate evolution by a factor of 100,000.
A number that frankly sounds impossible. It sounds like pure marketing hype until you actually dig into the methodology behind it.
And we will. And of course, we're going to look at the money. Where did billions of dollars move this month to shore up manufacturing capacity? And then there's the regulatory landscape that's, desperately trying to keep pace with all this innovation, especially with AI validation.
Which is rapidly becoming a massive bottleneck for a lot of companies, a huge one.
So let's start with the biology. Let's start at the source code.
Yeah.
Section 1 of the report, the biological blueprint. And we have to start with the synthetic yeast genome. On January 22nd, the consortium officially announced the completion of the final chromosome. This is the SC 2.0 project.
SC 2.0, yeah, this is monumental. To really frame this for everyone listening, this isn't just about tweaking a gene here or there with CRISPR. That's been standard practice for years.
This isn't just editing a Word doc.
No, this is writing the entire operating system from scratch. This is the first time we have built a complex eukaryotic genome from the ground up. We've done bacteria before, sure, Mycoplasma laboratorium years ago, but yeast is a eukaryote.
Which means it has a nucleus. It's organized like our own cells are.
Exactly. It has a nucleus, it has complex organelles, it shares a huge amount of its cellular architecture with human cells. That makes it an exponentially harder problem to solve.
And this project has been running for, over 10 years now. So my question is, why did it take so long? We've had gene synthesis tools for a while, we've had CRISPR. What was the actual holdup?
It boils down to two things, really. Stability and design complexity. You can't just, type out a DNA sequence in a text file, hit print, and expect it to boot up a living cell. It just doesn't work that way.
I wish it did.
We all do. The natural yeast genome is, for lack of a better word, a mess. It's full of junk.
Junk DNA.
Transposons, repetitive elements, introns, all this stuff that's been accumulated over millions of years of evolution. In a natural state, in the wild, these things are fine. They're part of the evolutionary toolkit. But for an industrial application, in a bioreactor, they are chaos agents.
No, though.
They cause mutations. They cause the strain to drift over time. Those transposons, the jumping genes, can literally copy and paste themselves into a new spot in the genome, disrupting a critical metabolic pathway you're relying on.
So you could have a star-performing strain on Monday, and by Friday, it's just average.
Or worse. It could be producing a toxic byproduct. The SE 2.0 team had to design a genome that was stripped of all that instability. They had to go through and systematically delete every single one of those transposons. They were essentially refactoring the code of life.
Like a programmer cleaning up messy legacy code.
It's the perfect analogy. And they had to do it while ensuring the cell could still function, could still divide, could still, live. It's like rewriting the operating system on your computer while you're still using it to send emails. It's an incredibly delicate process.
And beyond just removing the junk, They added features, right? The report mentions watermarks.
Right. They added specific DNA sequences that don't code for anything but act as a barcode, so you can always distinguish a synthetic chromosome from a natural one. It's a biosafety feature. But the really brilliant part, the part that's going to change everything, is the scramble system.
Okay, that's a mouthful. Break that down for us.
It stands for synthetic chromosome rearrangement and modification by LOXP-mediated evolution.
So it's an on-demand evolution switch.
It is an on-demand evolution switch. They've inserted these little genetic landing pads called LOXP sites all over the genome. Under normal conditions, they do nothing. But when you introduce a specific enzyme, it starts randomly shuffling the DNA between these sites. It creates massive genetic diversity on command.
So you can tell the yeast, okay, I need you to be more tolerant to ethanol,
Right.
and you just flick this scramble switch and screen for the survivors.
Precisely. You can evolve new traits in a matter of days that would take nature millennia. And then, and this is the crucial part, you can turn the switch off and lock that new, improved genome down again so it's stable for production.
So let's connect this to the factory floor, the so what for the industry. The report is very clear. It solves the reproducibility crisis.
It does. I mean, if you're running a fermentation facility, whether you're making insulin or a monoclonal antibody or a precursor for a biofuel, genetic drift is your worst nightmare. It's the silent killer of your profit margin.
You just lose yield over time.
You lose yield. You have a high-performing strain, you put it in a 50,000-liter tank, you run it for a few months, and by generation 50, it has mutated, its productivity drops, you get off-target metabolites that complicate your downstream purification. It's a constant battle.
And the synthetic genome is designed to be static. It doesn't drift.
It's designed for industrial rigor. It's a rock-solid production chassis. All the unstable elements are gone. What you put in the tank on day 1 is what you have in the tank on day 30. That consistency is gold.
Okay, so let's talk numbers. The report claims this enables 2 to 3 times higher yields. Where does that jump actually come from? Is the yeast's metabolism just inherently faster now, or is it more about efficiency?
It's both, but it's heavily weighted towards efficiency.
Yeah.
Think of all the energy in normal cell wastes. It's transcribing junk DNA, it's replicating transposons, it's running survival pathways that are totally unnecessary in a controlled bioreactor environment.
Just running all this background software you don't need.
Perfect. The SC 2.0 team deleted a lot of those non-essential genes. They also optimized the number of tRNA genes, which are crucial for protein synthesis. They've streamlined the whole process so that the cell isn't wasting metabolic flux, it isn't wasting ATP and carbon on things you don't care about.
So more of the sugar you feed, it goes directly to making your product.
Exactly. The metabolic flux is laser-focused toward the product of interest. This lets you run fermentations longer at higher cell densities without the culture crashing from accumulating toxic byproducts or exhausting itself. That's where the 2-3x comes from.
And that, in turn, impacts the facility footprint.
Oh, drastically. This is a CapEx game changer. If you can get 3 times the concentration out of a single batch, you don't need 3 50,000-liter bioreactors. You need 1.
Or you can run that one tank for a third of the time, freeing it up for the next product.
Right. It increases your facility's throughput. This lowers the capital expenditure barrier for entry for new companies significantly. It's a huge deal.
The report has this at a TRL 5, Technology Readiness Level 5. So validated in a relevant environment, but not yet full commercial scale. It says pilot commercialization is set for 12 to 18 months from now.
That sounds about right.
What's the biggest risk in getting from TRL 5 to, say, TRL 8 or 9? What could go wrong in the next 18 months?
Scale-up dynamics. It's always scale-up. A synthetic organism might behave perfectly in a 5-liter glass benchtop reactor where everything is perfectly mixed and controlled, but when you put it into a 10,000-liter stainless steel tank, the physics change.
How so?
You have hydrostatic pressure at the bottom of the tank that's immense. You have gradients in oxygen and pH and glucose concentration. The cell at the top of the tank is living in a different world than the cell at the bottom next to the sparger.
And we don't know if this highly optimized streamlined genome is fragile.
That's the billion-dollar question. Is it brittle? Does it pop when you stir it too hard? Can it handle the physical stress of industrial-scale processing? We just don't know yet. That's what the next 18 months of pilot runs will be testing, proving its robustness at scale.
Fascinating development. Let's pivot now to the other side of the design coin. Still in Section 1, but we're moving from the organism to the components. The evolution engine, T7 Oracle.
Right.
This came out of UC Berkeley early January reports, and the headline is just, it's a monster, 100,000x faster than nature. Now, I'm always skeptical of these huge exponential claims. Is this marketing fluff, or is the math actually real?
The math is real, but you have to be very careful about the baseline you're comparing it to. Nature is incredibly slow and inefficient.
It's working with random chance.
Totally random. Evolution relies on random mutation and then natural selection. It's a blind, stumbling walk through a vast landscape of possibilities. Even directed evolution, the technology that won the Nobel Prize that we've used for decades, is still essentially rolling dice.
You generate a massive library of mutants, you screen them all for a desired trait, you pick the winner, and then you repeat the process.
Exactly. And you are fundamentally limited by your screening capacity. You might be able to screen 1,000,000 variants in a high-throughput lab, but the potential number of protein sequences, what we call protein space, is functionally infinite. It's larger than the number of atoms in the universe. So you're sampling a tiny, tiny fraction of the possibilities.
It's hoping you get lucky.
You're hoping you get lucky. T7 Oracle is fundamentally different. It doesn't guess. It uses a large language model architecture, very similar to what runs GPT-4 or other advanced AIs, but it's been trained specifically on a massive data set of protein sequences and their corresponding functional data.
So it's not just navigating the fitness landscape, it's doing it intentionally. It has a map.
It has a map, and it's constantly updating that map. It creates a tight feedback loop. It uses the model to design a new sequence. It predicts the outcome, say, the enzyme stability at high temperature. Then it tests a small, intelligently chosen set of variants in the lab, feeds that new data back into the model, and the model learns. It gets smarter with every cycle.
So the 100,000x figure refers to the speed of traversing that design space to find an optimal solution compared to a random walk.
That's it, exactly. It's about the efficiency of the search. Instead of testing 1,000,000 random keys to find the one that opens the lock, it predicts the shape of the keyhole and crafts a key to fit.
The report specifically applies this to enzyme optimization and biocatalysis, and it calls out improving titers by 40 to 50% in continuous processes. Why the focus on continuous specifically?
Because enzymes in continuous flow systems take an absolute beating. They're not just sitting comfortably in a batch reactor. They are often immobilized, chemically stuck to a resin or a surface inside a column, and you have a constant high-velocity flow of substrate rushing past them 204/7.
So they degrade, they wear out.
They degrade, they unfold, they lose activity. The operational lifetime of your catalyst is a key economic driver in any continuous process. T7 Oracle was used to design enzymes that are hyperstable. They have stronger internal bonds, a more rigid structure. They simply don't unfold under that constant flow stress.
And that stability, that's the key that unlocks the non-sterile manufacturing angle mentioned in the bulletin.
This is the holy grail for industrial biotech. I mean, for decades, the assumption has been that you need perfectly sterile conditions, which is incredibly expensive.
Steam in place, validation, clean rooms.
All of it. It's a massive capital and operational expense. If you can run a process non-sterile, you can cut your OpEx in half. But to do that, your enzyme, your biocatalyst, has to be robust enough to outcompete any contaminating microbes that get in.
Or it has to work in conditions that kill those microbes.
Exactly, like very high salt concentration or extreme pH or high temperature. Most enzymes from nature would just fall apart in those conditions. T7 Oracle was able to design enzymes that not only survive, but thrive in those harsh industrial pockets.
So you're effectively hardening the biochemistry so you can soften the physical infrastructure around it.
That is the perfect way to put it. You trade computational cost and design time up front for massive capital and operational savings down the line. It's a paradigm shift. It's currently at TRL 4.5, but the potential is just enormous.
Let's move to the clinical side of Section 1 now. Early detection for Parkinson's. This is from researchers at Chalmers University of Technology in Sweden. They've identified biomarkers that can apparently detect Parkinson's years before physical symptoms appear, just through a blood test.
A huge clinical breakthrough, if it holds up.
Absolutely.
Right.
But we usually talk about manufacturing drugs, not diagnostics. Why is this specific news item in the Bioprocessing Bulletin?
Because of the sheer volume this implies. If this test gets through clinical validation, and it's already nearing it at TRL 6.7, and it becomes a standard screening tool for, say, everyone over 50, we are suddenly talking about manufacturing hundreds of millions of diagnostic assays annually.
And these aren't simple chemical scripts like a pregnancy test.
Not at all. These are complex immunoassays. They will almost certainly rely on things we make through bioprocessing. Recombinant antibodies, synthetic peptides, or specific enzymes needed to detect these biomarkers at very low concentrations in the blood.
So it instantly creates a massive biological reagent supply chain challenge.
The massive one. That's the direct manufacturing angle. But the deeper, more structural industry impact is on clinical trial design and success rates.
Right. The report mentions trial stratification.
Yes. Right now, late-stage clinical trials for Parkinson's drugs fail at a staggering rate. We're talking somewhere around 80 to 90%, billions of dollars down the drain.
And a big reason is that by the time you recruit a patient based on their symptoms, the neurological damage is already too advanced for the drug to make a difference.
That's one huge reason. The other is misdiagnosis. The patient might not actually have Parkinson's disease. They might have something that looks very similar clinically, like multiple system atrophy or Lewy body dementia, but which has a completely different underlying molecular driver.
So you put those patients into a Parkinson's drug trial.
And your drug looks like it failed, even if it was actually working miracles on the true Parkinson's patients in the cohort. The noise drowns out the signal.
So this biomarker allows for clean cohorts. You can ensure every single person in your trial actually has the disease your drug is designed to treat.
You reduce the noise to zero. The Bulletin estimates this could reduce clinical trial failures by 20 to 30%. Do you know how much money that saves the industry? We're talking tens of billions in R&D over a decade.
And that means more drugs actually succeed and reach the manufacturing stage.
Which in turn drives demand for the very manufacturing capacity we're always talking about. It's an upstream filter that unblocks the entire downstream pipeline. It's all connected.
Okay, let's move on from designing the biology to treating patients. Section 2 of the report, the nonprofit miracle. This is the approval of Woskera for Wiskott-Aldrich syndrome.
A truly brutal genetic immunodeficiency. Kids born with it have severe infections, autoimmune issues, cancers. It's devastating.
And this therapy, Woskera, was developed and pushed through approval not by a big pharma company, but by Fondazione Telethon, a nonprofit based in Italy.
An incredible achievement.
It is, and I want to challenge the standard narrative here a bit. We often hear that nonprofits and academic groups can do the early discovery, but they can't handle late-stage development because they just lack the capital for manufacturing. The costs are too high. How did Telethon circumvent the cost structure that big pharma claims is so necessary?
They didn't just throw money at the problem because they didn't have it. They attacked the process itself. They engineered the cost out of it through sheer efficiency.
Okay, explain that. Woskera is an ex vivo gene therapy.
Correct. The process is you take the patient's own hematopoietic stem cells out of their body, you use a viral vector, in this case a lentivirus, to insert a correct functional copy of the faulty gene, and then you infuse those corrected cells back into the patient.
And the most expensive, most difficult part of that entire process is making the viral vector.
By a country mile. Manufacturing clinical-grade lentivirus, or AAV, is incredibly inefficient. The yields are low, the purification is complex, and the cost per dose is astronomical.
And on top of that, you have the issue of transduction efficiency.
Right, which is the percentage of the patient's cells that actually take up the new gene when you mix them with the virus. Usually, in standard protocols, you're lucky to get 30 or 40%.
So to get a therapeutic dose of corrected cells, you need to start with a huge number of cells and use a massive amount of that very expensive virus.
You get it. It's a numbers game you're destined to lose on cost. The Telethon team focused obsessively on this one step, and through process optimization, they managed to achieve 85 to 95% transduction efficiency.
That is a staggering jump. That's not an incremental improvement. How did they do it?
The full details are proprietary, of course, but it's likely a combination of a few things. They probably developed a cocktail of transduction enhancers. These are small molecules that essentially grease the wheels and help the virus enter the cell more effectively. They also likely spent years optimizing the cytokine cocktail used to keep the stem cells healthy and receptive during the process.
So by hitting that 95% efficiency,
By hitting 95%, the whole economic equation flips. You need far less virus per patient, which drops the cost. You can start with fewer of the patient's cells, which is less taxing on them. You shorten the time the cells have to be kept alive outside the body, which reduces the risk of contamination or differentiation. You reduce the failure rate of the entire manufacturing batch.
so they didn't just fund the therapy, they fundamentally re-engineer the cost out of the process.
They proved that process intensification can be a direct substitute for massive capital scale. You don't need a billion-dollar commercial footprint if your core unit operations are running at near-perfect efficiency.
Which is a huge lesson for the entire cell and gene therapy space. But let me play devil's advocate. Is this model truly scalable? A nonprofit curing a rare disease where the entire patient population is a few hundred people worldwide is one thing. Can this telethon model work for something like sickle cell anemia, where you have tens of thousands of patients waiting?
That is the big debate right now. The telethon model, as it stands, works for ultra-rare diseases. For larger indications, you still face a colossal logistics challenge. You need apheresis networks to collect the cells, a robust cold chain to transport them, massive data management systems to track every single patient's batch. A nonprofit might struggle with that global logistics layer.
Which is what Big Pharma excels at.
It is. But the science, the breakthrough in achieving high transduction efficiency, that is completely transferable. Every cell therapy company, Big Pharma included, should be looking at this WASGERA data and asking their own process development teams, why are our commercial processes lagging so far behind a nonprofit's efficiency?
It's a wake-up call on the importance of process development, not just the initial discovery.
It absolutely is. And this isn't a theory. It's TRL9. It's done. It's approved. It works.
All right, let's take that idea of process efficiency and move it to the broader factory floor. Section 3, revolutionizing the factory floor, all about process engineering. And the big theme here is the final, decisive shift from batch to continuous.
We've been talking about it for years, but now it's really happening.
The data seems to back that up.
Yeah.
Late 2025 surveys cited in the Bulletin show that 60% of firms are now actively planning to implement multi-column chromatography.
The tide has well and truly turned. For years, the hesitation was all about risk. The question every operations manager asked was, if I'm running a continuous process for 30 days straight and I get contamination on day 10, do I have to throw out the entire run, the entire 30 days worth of product?
Which would be a financial catastrophe.
A complete disaster. But the technology has matured, the control systems, the inline sensors, the analytics. They've gotten so good that we can detect and divert problems in real time. The perceived risk has dropped below the threshold of the obvious efficiency gains.
Okay, so let's get specific on multi-column chromatography. Walk us through the benefit here. Is it just about going faster?
Speed is part of it, but the bigger wins are in resin utilization and buffer consumption. Let's think about traditional batch chromatography. You have one giant expensive column packed with resin.
And protein A resin for monoclonal antibodies is not cheap.
It's famously called liquid gold. It can be thousands of dollars per liter. In a batch process, you load your unpurified product onto this giant column, but you have to stop loading before the resin is fully saturated.
Why is that?
Because you're afraid that if you load too much, your valuable product will start to leak out the bottom before it has a chance to bind. So to be safe, you only use maybe 60 or 70% of the resin's total binding capacity. You're wasting a third of your most expensive consumable.
That's incredibly inefficient.
It is. Now, in multi-column continuous chromatography, you have a series of much smaller columns. You load the product onto the first one. As soon as it's 100% full, any overflow is automatically diverted to the second column in the series. While the second is filling, the first is being washed and eluted.
So you're using 100% of the resin capacity all the time.
All the time. And because you are cycling them so quickly, the columns can be much, much smaller, which is another huge cost saving. This leads directly to the other major point in the report: a 40% cut in buffer use.
And buffers are the hidden killer in a facility's operating expense.
They absolutely are. It's not just the cost of the chemicals. It's the cost of the water for injection, the WFI needed to make them. It's the space taken up by massive buffer prep and hold tanks. It's the labor for testing and releasing every single batch of buffer. If you can cut your total liquid handling volume by 40 or 50%, your entire facility footprint shrinks.
Speaking of shrinking footprints, that's a perfect segue to the next topic here, the honey I shrunk the bioreactor moment. Novel capillary fiber systems.
This is a radical departure from the stirred tank reactor we've been using for half a century.
Right. For 50 years, the basic model for making biologics has been to put cells in a giant steel tank and spin a propeller, an impeller, to keep everything mixed.
Which works, but it has a fundamental physical limitation: shear stress.
The force of the liquid flow in the spinning impeller can literally tear the cells apart.
It damages the cells, which limits how dense you can pack them in. You reach a point where if you spin the impeller any faster to improve mixing, you just kill the cells. Capillary fiber systems completely eliminate that problem.
So how do they work? The report says it mimics the human circulatory system.
That's a great way to think about it. Imagine a bundle of thousands of tiny, hollow fibers, like microscopic straws. The cells are grown in the space outside the fibers, the extra capillary space. They pack in together incredibly tightly, almost like a real tissue.
And the nutrients.
The nutrient-rich media flows through the inside of the hollow fibers and then diffuses out across the fiber membrane to feed the cells. Waste products from the cells diffuse back into the fibers and are carried away.
So there's no impeller, no shear at all.
Zero mechanical shear. And because the diffusion distance for nutrients and waste is so short, just a few micrometers, you can reach cell densities that are 10, 20, even 50 times higher than what you could ever achieve in a stirred tank.
Which is how you get to the headline stat. A 50-liter setup can now match the output of a traditional 5,000-liter fed batch tank.
The mass transfer limitations are virtually gone. You're creating a perfect little artificial organ, and the energy implication mentioned in the bulletin, a 30 to 50% reduction, is huge. You aren't paying to heat, cool, and spin thousands of liters of liquid anymore.
Okay, so this sounds revolutionary. Why isn't everyone throwing out their steel tanks and buying these? If I can replace a warehouse-sized fermentation suite with a room full of these 50-liter units, what's the catch?
The catch is that the stirred tank, for all its flaws, is a known beast. We have 50 years of data on it. We know how to control it. We know how to model it. We know how to validate it for the regulators.
And these new systems are more of a black box.
A bit? How do you get a truly representative sample from a packed bed of cells that's as dense as tissue without disrupting the whole system? It's difficult. And the biggest operational risk is fouling. If those tiny capillary fibers get clogged, you're done. The whole run is dead and you can't save it.
So the operational risk is currently higher, even if the physics are superior.
For now, yes. The control systems are still catching up. But for certain applications, like point-of-care manufacturing, imagine making a personalized cancer vaccine right there in the hospital basement. This technology is the absolute future.
Let's quickly touch on the last part of this section, the digital twins case study. A company called Exozymes, they're reporting 99% conversion and 91% yield in an enzyme-based process. The key, they say, was using AI for PAT.
Right, for process analytical technology. This is all about closing the feedback loop in real time.
Traditionally, PT involves putting sensors in your reactor, Raman spectroscopy probes, pH sensors, dissolved oxygen probes, and they spit out a bunch of data.
And an operator, a human, looks at that data on a screen and maybe makes an adjustment to a feed pump every few hours. It's a reactive process. Exozymes connected those sensors directly to an AI control model, the so-called digital twin.
So the AI is the one turning the knobs, not the human.
It's doing more than that. It's predicting where the process is going to be in the next hour. Based on the current trend in glucose consumption, for example, it can predict that the cells are about to experience stress. So it adjusts the feed rate proactively before the stress event ever happens.
It's keeping the process aggressively in that perfect Goldilocks zone of peak performance.
Constantly. That predictive capability is what gets them from a good yield to a near-perfect 91% yield. It prevents the small deviations that, over time, lead to a culture crashing.
The report says that industry-wide, just implementing these kinds of digital overlays is boosting yields by 15 to 20%.
And that's without buying any new steel. It's just running your existing facility with better code. It's a massive win for efficiency.
Okay, this is a good point to shift gears from the factory on Earth to, well, the factory off planet. Section 4, Biomanufacturing in Space. Fact versus fiction. And the big news here is the BioStar pilot.
Size. Okay, let's talk about space.
I hear that sigh, I hear the skepticism, but let's look at the data first before we get into the reality check.
Yeah.
The results came down on January 27th. It was a space-based, cell-free biomanufacturing experiment.
On the space station.
And the result was that it converted waste streams into methane with 80% efficiency. The report says this signals potential for 2 to 3 times productivity gains in these kinds of closed-loop systems.
Look, the data is impressive. I am not, for 1 second, disputing the result. The science is sound.
Okay, so explain the science. Why is it more efficient in space? What does microgravity actually do to the chemistry of a cell-free reaction?
It eliminates 2 fundamental physical forces that plague us on Earth: sedimentation and thermal convection.
Meaning things don't sink and heat doesn't rise.
Exactly. On Earth, if you're trying to mix enzymes and substrates in a liquid, the heavier components will eventually start to sink. The reaction vessel will heat up, creating convection currents that cause chaotic fluid movement. You get gradients. The concentration of reactants isn't uniform.
So you have to constantly stir it to keep it mixed.
And stirring adds shear force, which can damage the delicate enzymes. In microgravity, on the other hand, diffusion is the dominant force. Everything stays perfectly suspended, perfectly distributed. You get these beautiful, uniform reaction kinetics that are almost impossible to replicate on a large scale on Earth.
So for a cell-free system, where you just have a soup of enzymes and substrates, it's the perfect mixing environment without the violence of mechanical mixing.
Correct. And that's why they saw those 2 to 3 times productivity gains. The reaction was just cleaner, more efficient at a molecular level.
Okay, so here's the argument from the space hype crowd. If we can get 3x productivity, why aren't we launching orbital factories? You mentioned in the report that the terrestrial infrastructure costs are a barrier.
Let's just do the basic unit economics. To get 1 kilogram of payload to low Earth orbit, even with the cheapest rockets like Starship, you're talking thousands of dollars. Then you have to operate the automated factory in space, and then you have to bring the finished product back down through the atmosphere, which is another costly and risky step.
Right.
The 3x productivity gain you get in orbit does not come close to offsetting the, say, 1,000x cost of logistics for a commodity product like methane or even a standard monoclonal antibody. It just doesn't make sense.
But what about for very specific high-value products?
Yeah.
The report mentions things like growing perfect protein crystals for drug design or complex tissue engineering.
Now that is the actual viable niche. If you are making something where absolute perfection and the absence of gravity is the enabling factor, like a protein crystal that needs to be flawless for X-ray crystallography to determine its structure, or printing a human heart where you need the layers of cells to assemble without collapsing under their own weight, then space wins.
But for bulk biomanufacturing,
For making mass quantities of a drug that we already know how to make on Earth, it makes infinitely more sense to spend that $1 to $2 billion on upgrading our terrestrial factories with those capillary fiber systems and digital twins we just discussed.
we can try to mimic the effects of microgravity here on Earth.
To a certain extent, yes. We have technologies like rotating wall vessels, acoustic levitation. We can get close enough to reap most of the benefits without having to deal with the rocket equation.
And then there's the regulatory aspect you flagged. This is fascinating. The jurisdiction issue.
It's a complete legal black hole. Imagine this scenario. A French company develops a drug. They launch their cell-free reactor on an American rocket from U.S. soil. The drug is manufactured in international space aboard the station, and then it's brought down and sold in Japan.
Okay.
And then a purity issue is discovered. Who is liable? Which set of good manufacturing practice, or GMP, standards apply? The FDA's, the EMA's, Japan's PMDA.
You'd need a Space Force division of the FDA.
We can laugh, but the Bulletin cites compliance experts who estimate the cost of just managing that cross-jurisdictional paperwork would be 15% higher than for a terrestrial product. And for any commercial manufacturer, that level of legal uncertainty is absolute poison.
So the bottom line on BioStar is that it's a brilliant proof of concept for enabling long-duration missions to Mars, but it's not a replacement for the manufacturing hub in New Jersey.
That's it, exactly. It solves the how do we make medicine on Mars problem. It does not solve the how do we lower the cost of insulin in Chicago problem.
Let's follow the money back to Earth then. Section 5. Follow the money, looking at industry moves and investments. And the big story this month seems to be the continued rise of the CDMO.
The consolidation phase is in full swing.
The report frames this in the context of the looming patent cliff. We're talking about an estimated $300 billion in blockbuster drug revenue going off patent in the next few years. Why does that specifically drive the growth of CDMOs, the contract development and manufacturing organizations?
Because Big Pharma is panicking. It's a race against time. When you lose patent exclusivity on a drug that brings in $10 billion a year, your revenue plummets. You need to launch new drugs fast to fill that massive gap.
And building your own new manufacturing facility takes, what, 4 to 5 years?
At least. From groundbreaking to validation. But signing a contract with a ready-to-go CDMO, that takes 4 to 5 months.
So you're not just buying capacity, you're buying speed to market.
You're buying speed and flexibility. If your new drug fails in phase 3, you're not stuck with $1 billion empty building. You just terminate the contract. It de-risks the pipeline. And that's why we're seeing these huge deals. Avid Bioservices was acquired by private equity for $1.1 billion. That's a massive premium. The buyers, GHO and Ampersand, are making a huge bet that the demand for outsourced capacity is going to far outstrip the supply.
Then there's the Lonza deal. They're in the process of buying a Roche facility in Vacaville for $1.2 billion. This one is interesting to me. They're buying a legacy site Usually you want the shiny, new, state-of-the-art facility. Why buy an old Roche plant?
Because it has the bones, it has the utilities, it has the water purification loops, the power grid connections, the waste treatment, and most importantly, it has the environmental permits. The shell is there, and it's already approved.
So they're going to gut it and renovate.
Exactly. The Bulletin says Lonza's plan is all about upstream intensification. They're going to rip out the old, giant stainless steel tanks and put in modern high-density perfusion systems, or those continuous processing skids we just talked about.
They're essentially flipping the house, or flipping the factory.
They're flipping the factory. They believe that they can run that site 15 to 20% cheaper than Roche could because Lonza's entire business is manufacturing operations. Roche's business is drug discovery. It's a classic case of division of labor and core competencies.
And the third deal mentioned, Bora Biologics buying an emergent biosolution site for only $30 million.
A very different kind of deal. This is for expanding their ADC manufacturing.
Antibody drug conjugates. These are the super potent cancer drugs that are basically a guided missile.
Right. An antibody that seeks out the cancer cell linked to a highly toxic chemotherapy payload. Because the payload is so potent, you need specialized high-containment facilities to manufacture them safely. You can't just make these in a standard monoclonal antibody plant.
So Bora, picking up a site for just $30 million, is a steal.
It's a distressed asset sale. That site had issues, and Bora is getting it for pennies on the dollar, betting they can fix it up and turn it into a valuable asset for the booming ADC market. It highlights the bifurcation in the market. High-value, flexible assets go for billions. Specialized distressed assets go for a fraction of that.
The report's synthesis is that these moves signal a 25 to 30% sector consolidation over the next 1 to 2 years.
I think that's right. The big players are buying up capacity rather than building it from scratch. For you, the listener, if you're at a smaller biotech, this means you might have fewer CDMOs to choose from in the future, but the ones that remain will likely be larger, more robust, more technologically advanced partners.
Okay, let's look at the other side of the money equation, the startup ecosystem and R&D expansion. The report flags that startups from Washington University hit a cumulative $1.7 billion in funding, and Genentech is in the middle of a $2 billion expansion in North Carolina.
The Genentech expansion is the one to watch closely. A $2 billion investment is a huge vote of confidence in the industry. But the key phrase in their press release, which the Bulletin highlights, is that they are targeting labor efficiency gains of 20%.
That sounds like corporate speak for, we are planning to hire fewer people.
It is. But it's not because they want to fire people or cut jobs. It's because they can't find the people to hire. The talent shortage in this field is becoming acute.
Really? In what areas?
Bioprocess engineers, automation specialists, quality control, and validation experts. The 2025 jobs report was a massive red flag. We are simply not graduating enough people with the right skills to run these new, highly complex, automated facilities. So Genentech is designing the factory from the ground up to be able to run with fewer operators because they know the operators they need simply don't exist in the labor pool.
That is a major bottleneck, and it leads us perfectly into the next section, Section 6, the rules of the game. Let's talk about the regulatory landscape.
The invisible hand that can speed everything up or grind it to a halt.
And the big news was on January 14th. The FDA and the EMA, their European counterpart, jointly released new principles on the use of AI in drug development and manufacturing. The key phrase seems to be risk-based validation.
This is the regulators basically saying, okay, we get it. We know you want to use AI and machine learning for everything, but you can't just treat these models like a black box and tell us to trust you.
What does risk-based mean in practice? Give me an example.
Okay. If you use an AI model to optimize your warehouse inventory for lab supplies, that's a very low risk. If the AI hallucinates and gets it wrong, you just end up ordering too many beakers. No one gets hurt.
Right.
But if you use an AI model to determine the final release criteria of a drug batch, if the AI is making the final call on whether a batch of medicine is safe to be injected into human beings, that is the highest possible risk.
So for that high-risk application, the validation requirements are going to be immense.
Exactly. You're going to have to validate the model's architecture, the quality and integrity of the data it was trained on. You'll have to show how you monitor it for drift over time. The Bulletin says a 95% compliance documentation requirement is expected. The paperwork burden just exploded for anyone using AI in a critical GMP context.
At the same time, we're seeing initiatives to speed things up. The FDA is launching a pilot program called PreCheck.
I love the branding on that. Sounds like airport security.
It does. It's launching February 1st, and the promise is that it will cut GMP inspection delays by 30 days for companies that have a good track record.
Now, 30 days might not sound like a lot to an outsider.
But in a drug launch sequence, it's everything.
It's everything. If you're a pre-commercial biotech and you're burning cash at a rate of $500,000 a day while you wait for that final approval, getting to market a month early is a $15 million swing. Not to mention, it gets a potentially life-saving product to patients that much faster. It's a big deal.
But there's always a trade-off. The report points out that while the EMA's prime scheme for accelerated assessment can reduce review times to 150 days, it also requires 20% more data on biosafety upfront.
Faster review does not mean easier review. In fact, it usually means the opposite. It means a compressed review. You have to have your entire data package completely locked down and perfect before you even apply. Your biosafety data viral clearance studies, impurity profiles, has to be rock solid. There's no room for error.
Let's talk about the acronyms that make people in this industry sweat. The report goes into the nuances of change control, specifically CBE 30 versus PAS.
The change control nightmare, the bane of every manufacturing scientist's existence.
For the uninitiated listening, explain the pain here. Why do these acronyms matter so much?
Okay, so you're running a validated manufacturing process in your factory. You've been doing it for years. One day, your team realizes that if you switch from filter supplier A to filter supplier B, you can improve efficiency and save the company $100,000 a year.
Seems like a no-brainer.
You'd think so. If that change is deemed minor and has no potential impact on the final product's quality, you can file it with the FDA under a CDE 30. That stands for changes being effected in 30 days. You tell the FDA what you're doing, you wait 30 days, and if they don't object, you're free to make the change.
Okay, that's manageable.
But, and this is a huge but, if the FDA decides that this new filter might have an impact on product quality, even a tiny one, it gets classified as a PAS. A prior approval supplement.
Oh, that's bad.
That means you have to conduct new studies, generate a full data package to prove the change is acceptable, submit it to the agency, and then wait for them to review it and give you explicit permission. That process can take 4, 6, sometimes even 9 months.
So your $100,000 a year improvement might not even be worth implementing because the regulatory wait time and the cost of the studies eats up all the savings.
Exactly. It freezes innovation on the factory floor. People become afraid to make common sense improvements. The new guidelines from the ICH Q13 are trying to clarify this for continuous processes, but the expert insight in this month's bulletin suggests that, if anything, post-approval changes are getting tighter, not looser.
Why? Because the new, complex processes are more interconnected.
That's the theory. With a highly integrated continuous process controlled by AI, a small tweak in one unit operation can have unforeseen ripple effects downstream that are hard to predict. So the regulators are, understandably, being extra cautious.
All right, let's move to Section 7, Innovators to Watch. A look at some of the startups that made news in January, and we have 3 very distinct flavors here.
Yeah, we have infrastructure, formulation, and then the far-out longevity play.
Let's start with Synonym Biotechnologies.
Yeah.
They were in the latest Y Combinator batch, and their whole pitch is about building fermentation infrastructure.
Think of them as the Amazon Web Services for biology.
Okay, I like that.
AWS built the massive standardized server farms so that any software developer could rent computing power by the hour instead of having to build their own data center. Synonym is trying to do the same thing for biomanufacturing. They're building large-scale, standardized fermentation facilities.
So a food tech or a materials startup can just rent tank time from them?
Exactly. They can scale up their new strain without needing to raise $50 million to build their own pilot plant. Synonym claims their AI-optimized, standardized designs can cut the cost of goods for these products by 30 to 40%. It's a platform play, and if they can pull it off, it could unlock a whole wave of innovation in commodity biologics.
Next up, Persist AI. They are focused on automating formulation.
Formulation is often called the dark art of drug development. It's a huge bottleneck. You have this amazing new drug molecule, but how do you actually get it into the body in a stable, effective form?
Does it need to be a pill? An injection? Does it need to be wrapped in a lipid nanoparticle to protect it?
Right. And usually this is figured out through a very slow, manual trial and error process by scientists with pipettes, testing one recipe at a time. It can take years.
And what is Persist AI doing differently?
They're using a combination of robotics and machine learning to test thousands of different formulations in parallel in a high-throughput way. Their specific focus is on sustained-release drugs formulations that can make a drug last for 30, 60, or 90 days in the body from a single injection.
That's a huge deal for patient adherence. Nobody likes taking a pill every single day.
It's a game changer for adherence, especially for chronic conditions. And for complex molecules like peptides, which degrade very easily in the body, finding the right protective formulation is incredibly difficult. Persist is essentially brute forcing the problem with AI and automation, and they claim they can cut that development time by 50 to 70%.
Very cool. And finally, the high-risk, high-reward play. Rejuve Biotech. Their focus is on longevity therapies using AI-driven epigenetics.
This is the moonshot. They're trying to do nothing less than reverse the aging process at a cellular level.
The Benjamin Button strategy.
In a way, yes. They're using AI to map the complex epigenetic markers, the chemical tags in our DNA, that change as we age, and then they're developing drugs to try and reset those markers back to a more youthful state.
They've got over $10 million in backing.
Which, in the longevity space, is really just seed money to get started. Their biggest challenge isn't the science, it's the clinical translation. How do you run a clinical trial to prove you've slowed down aging? The FDA doesn't recognize aging as a disease. Do you have to run a trial for 30 years and measure lifespan?
It's not practical. Unless you could use some of those validated biomarkers we talked about back in Section 1.
Everything connects. That's their path forward. If they can get the FDA to agree on a validated surrogate biomarker for aging, maybe a specific panel of epigenetic clocks, they could run much shorter trials and get a much faster readout. But it's still a very high-risk venture.
Okay, Section 8. Let's do some critical analysis and look ahead. It's time to land this plane. We've covered a huge amount of ground this month. What's the grand synthesis? What does it all mean?
The synthesis, for me, is one word. Maturation. The whole sector is finally maturing. We're moving past the science project phase where you'd have these cool ideas that worked in the lab but could never scale or be made economically.
Valley of Death.
The valley of death. What we saw in January is that technologies like AI and continuous processing are finally starting to fix the fundamental unit economics of biology. We are seriously addressing the 20 to 30% yield losses that have plagued the industry for decades due to batch variability and process inefficiency.
But the bottlenecks are shifting. They're becoming human.
The bottlenecks are now human and logistical. That talent shortage we talked about is not a joke. That 2025 jobs report was a blaring red flag. We are desperately short on bioprocess engineers, on automation experts, on regulatory and validation specialists.
So if we don't fix that talent pipeline, all these shiny new $2 billion factories will just sit idle or run well below their potential capacity.
Or they'll be run inefficiently. The single issue could delay the scale-up and adoption of all these new technologies we've discussed by 12 to 18 months. And at the same time, the supply chain for critical consumables, single-use bags, filters, chromatography resins is tightening again. The Bulletin warns we could see shortages rise by another 10 to 15% this year.
So with that in mind, what are you watching for specifically in February 2026?
3 things. First, on February 15th, the EMA is holding a Q&A workshop on their post-approval change protocols. I'll be listening very closely to see if they signal any new flexibility on that CBE 30 versus PAS issue. If they make it easier to innovate on the factory floor, the industry will breathe a huge sigh of relief.
Okay, number 2.
FDA delays, PDUFA dates. The report mentioned the Novavax vaccine decision was missed in January. I'll be watching to see if that was a one-off or if other decisions start slipping. It's the canary in the coal mine that tells us if the agency is getting overwhelmed by the complexity of these new applications.
And finally,
The YC Industrial Bio Cohort demos at the end of February, I want to see how many of those new startups are making claims about enzyme productivity. If we see 2 or 3 new companies armed with tools like T7 Oracle hitting those 2-3x productivity jumps, it confirms that this trend is real and accelerating.
it has been a wild month. We've gone from designing yeast from scratch on a computer to shrinking entire factories down to the size of a fridge and even printing medicines in Earth orbit.
It has. It's been incredible. But I'll go back to my opening point. The real victory this month, the thing that really matters, isn't the flashy sci-fi headline.
It is the boring details.
It's the boring details. It's the 20% yield boost from a digital twin. It's the 30-day inspection cut from the FDA pre-check program. That is the stuff that actually makes this incredible future affordable and accessible for everyone.
It's the quiet triumph of boring efficiency over flashy, expensive inefficiency.
Amen to that.
So here is the final provocative thought I want to leave you with, and it ties everything together. We discussed T7 Oracle, an AI that can evolve proteins 100,000 times faster than nature.
Right.
We discussed digital twins, AIs that can run physical factories with predictive control. We discussed Synonym, a company building standardized automated infrastructure.
I think I know where you're going with this.
Are we approaching a point where biology stops being a manufacturing problem, a problem of steel and steam and pipes, and becomes purely a software problem?
Yeah.
If we can code the cell's genome from scratch, simulate the entire process in a digital twin, and deploy it on automated standardized hardware, does biology just become another form of code?
That is the trillion-dollar question, isn't it? If it becomes code, the pace of innovation can accelerate exponentially, following a Moore's law-like curve. If it remains stuck in the messy physical constraints of biology, we stay on a slower, linear path of improvement. What we saw in January 2026 suggests that the entire industry is trying very, very hard to go exponential.
A fascinating month and a fascinating thought to end on. Thanks for diving deep with us.
Always a pleasure.
That's this month's Bioprocess Intelligence. If you're turning biology into manufacturing at scale, these are the signals to watch. I'm Prasad Ernala. See you next month on Biomanufacturing and Fermentation Technology.